medical research career. So he's going to talk to us about the different things that he's worked on and what he's doing now. And if you all have questions, just hold up your hand and ask, and he will stop and answer them. So, Dr. Kotwal, it is yours.
Okay. Good afternoon, everyone. Sorry for the delay. Things beyond my control a little bit. And oh, can you hear me back there? Okay, great. Thanks. So I'll be talking about mainly about dealing with Alzheimer's disease. Most of you are probably familiar with it, and hopefully you don't have any of your grandparents with any of this condition, but there are at least 6 million around the country that have Alzheimer's, and it's a very difficult disease. Although there are good signs that things may be improving in the future. There are 2 FDA-approved drugs, a lot of side effects, and they're not delivered the way we are going to deliver our protein. And so hopefully there should be a better future for people with Alzheimer's disease. So, is there a pointer?
No, I didn't think to have one. I'm sorry.
That's fine.
It doesn't work on the TV anyway.
Oh, okay. But maybe I
can use your mouse.
Oh, yes. Okay. So basically, most of you are probably generally familiar with one of the most critical organs of the body, the brain. And it is very well mapped into regions which have specific functions. And one of the functions that I'm interested in is where the memory and the other important aspects of cognition, understanding time, and things like that are located. And that region would be around here. It's also called the limbic system. So what I'm going to talk about is what happens in the case of Alzheimer's disease and how we are going to try to develop a treatment so that if somebody has done a genetic analysis of their DNA, they will know that they are predisposed to Alzheimer's disease. And if they know that, then they can act very earlier on during that period. So you don't have to wait to be hit by it and it's too late. So this way, even before the onset of Alzheimer's, you can slow the process down and live as though you're not going to lose your mind, because mind is a terrible thing to waste. So it's very important to have to protect your memory. And you will realize when you reach my age that memories are very precious. And that's what keeps you going. That's what gives you purpose in life. And if you lose that, you're lost yourself, basically. And so that's something that we're trying to prevent here. So I started with genetic engineering in pox viruses. Have you heard of pox viruses? If not, I guess most of you are born before there was smallpox. When I was born, smallpox was still around. But in 1978, smallpox was declared dead. And it was declared dead because there was no animal host for it. Like you have hosts for COVID, you have hosts for other organisms. But with smallpox, human beings were the only host. And all the human beings were vaccinated by the time the smallpox was declared dead in 1978 by WHO. But it is a large virus, and so because it is a large virus, it had the capacity to interact with the host and to suppress the immunity that is directed at the virus. So that was what made that. And in history, probably the smallpox was the one that killed the most number of people. And it took Jenner, the vaccination was in 1700s, 1796, and it took 180 years to get a vaccine. So we should be fortunate that the COVID vaccine was, did not take that long. The warp speed or whatever speed, they just had the vaccine, but it's not eradicated. Smallpox is eradicated, whereas COVID goes through generations. Every 60 million there's a new variant, and now they've run out of the Greek alphabets after Omicron, and they're calling them subvariants. So that is a long way to get those variants. Whereas smallpox did not mutate a lot, but when it infected it, basically 30% of the people were killed by the smallpox virus. So you must have heard about gain of function, how this COVID arose or emerged from nowhere and caused the pandemic. How many of you have heard of gain of function? Gain of function is when you engineer a virus to gain a new function. And normally that's done only for studies. It's not a very good thing considering public health cost of what has happened. More than 6 million people have died, assuming that it happened due to gain of function, that when it was engineered, it became more virulent. I was interested in doing the opposite. I was interested in taking away the virulence. Loss of function was my goal, and that was easy to engineer new viruses by removing the virulence genes and then putting in, and then the virus became attenuated. Because even though I talked about the smallpox virus that was used in the eradication, the vaccinia virus that was used for eradication, the virus that first was developed by Jenner, the virus that was used for eradication was still kind of caused complications. It was not 100% safe. And so to remove the virulence genes and make a virus that is much more attenuated, we use the word taming of a virus, was something we wanted to use as the thing. So that's the genetic engineering, and I will tell you, we engineered several viruses and removed the virulence genes, and then checked whether this could be used as a future vaccine against smallpox or monkeypox. Monkeypox, some of you may have heard, has been around now in humans. Originally, it originated from in the Democratic Republic of Congo, and then it moved over to humans, and then transmitted from humans to humans. But then again, the same vaccine that was used against smallpox is useful against monkeypox. So from there, I found one protein that was attributed to virulence, and that gene was then engineered to make a new, into yeast to express a large quantity of the protein. And the reason for that is this protein was able to block inflammation. And so, and inflammation can happen several ways. But this inflammation we are talking about is the inflammation that is due to, in the brain. So this is what, or in the spinal cord, or in the, after head trauma, or after Alzheimer's, there is inflammation. So that was what the engineering of that. But engineering in a lab is not the same as manufacturing. So we have to really manufacture it on large quantities so that we can do the human studies and those things and all that. So this protein that we are interested in is what will do the downregulation of the neuroinflammation, which actually causes the brain damage that results in loss of memory and other features that we don't want the brain to lose. So this has been a quest from the time of doing genetic engineering to trying to now prevent Alzheimer's disease in those who know their genetic composition in terms of our genetic sequence in terms of the Alzheimer's disease. There are certain genes which predispose a person to Alzheimer's disease. And what we are trying to do is to first target those people who are predisposed, and predisposition, you don't have to wait for them to be in their old ages to find out whether they had this family history or things like that. You can find out at birth. But I recommend that by age 55, everyone should try to know whether you have that particular predisposition so that you can be proactive in preventing memory loss in later years. And so that is the ApoE4 protein. ApoE4 protein is encoded by the ApoE gene, but it has 4 alleles. What are alleles? Alleles are alternative forms of a gene. So you have one gene, ApoE, but it has 4 alternative forms based on differences in sequencing. So out of the 4, most people who have ApoE2 or ApoE3 have normal genes. So they are not going to be predisposed to Alzheimer's. But if one has ApoE4 allele, then they are predisposed to Alzheimer's disease, 2 to 3 times more than the rest. So there are several risk factors. Aging is the biggest risk factor, but having the ApoE4 allele is also a big risk factor. But that doesn't mean it's the end of the world. That's because advances are happening, such as the ones that I'm going to talk to you about. But also, the ApoE4 is also connected with, interlinked with diabetes and heart disease. So one needs to take care of all of that and then try to reduce inflammation in the brain, and then that will basically. So I basically told you the objectives of my what I'm trying to tell you, and now let's move on to. Okay, I have to disclose I do have conflicts of interest. I do, of course, personally, I would like to, from the rest of my life, I want to make sure that I have delivered an early treatment for Alzheimer's disease. So I'm co-founder and president and CEO of Inflamed Incorporated. Inflamed was formed along with a for-profit entity of the University of Louisville, had a entity called Metasite, and that Metasite was there to spin off innovations from the university. So that entity was basically involved in that. But they took a chunk of the entire 100% equity to get you started, basically hire a lawyer and form the company and all that. I see a lot of young people nowadays. A lot of the youngsters are starting companies, and so took me a while, but for you can just start your companies as soon as you have an idea, because to translate that idea, it takes a while. You need money, and the government will give you money. Like here's Dr. Koenig, who has a whole lot of funding from the government to do innovative research, but he has worked all his life to get there. But somebody starting off can get that funding too. So then Metasite folded and is now managed by the University of Louisville Research Foundation. So University of Louisville Research Foundation has an equity in Inflamed, the company that that we co-founded together with the Metasite. So one innovation center is on the corner of Brook and Jefferson in downtown, and where you see University of Louisville, third floor. Below that is my office, and it's a great wet labs incubator, which means startups. You don't have all that capital to start a company and so on. So you don't have to have a lot of money. You have an idea, you have a mission, you have a purpose. You find an incubator, and then you start developing your ideas and apply for funding from the government, and SBIR funding, small business innovation research funding, and then you will have investors interested in funding you, and so that's what will take you forward. So the SBIR is sort of a good start for doing that. Now, the history about this place is very interesting. Initially, it was called Nucleus, and there was a company called Potentia, and Potentia was looking for ideas. And I happened to meet the CEO in the elevator, outside the elevator at the university. I was an academician then, and not very much into business. Didn't know I needed to patent my ideas or whatever. So I just gave him the idea of how to target complement system in the body, a powerful system, to develop new treatments. And that guy ran with the idea, and he was trying to use that idea for macular degeneration. And in doing so, he made some advances, and then he sold the company and started another company which is now traded on Wall Street called Apellis, and that is for eye-related problems that he was dealing with. And then he went from there. He headquartered in Crestwood, which is Oldham County, and from there he is now headquartered in Boston. So a company that started with pretty much just an idea is now valued at $2 billion. And its stock value today is 4 times the stock value of a company that has been around for over 100 years, Ford Motor Company. So there is funding to do that, because what we are trying to do is okay to start with $500,000 and all that, but ultimately you have to go through the FDA process. You need at least $100 million to keep doing. And so the valuation of the company of that is $2 billion, but it's that whole thing started in this place. So that's pretty historic, and hopefully what we accomplish will also. And then there's another small business there that is interested in working on lyophilized blood. So blood transportation to the battlefront or to the hospitals will be much easier to do. So that is also in one of the labs. There are several labs, and the good thing about this is you are committed only for a year. So whatever you can accomplish and then move to a bigger facility, whatever, just like Potentia did, you can do it. But it's initially that they have equipment, and it's very nice conference rooms and everything. In fact, I had invited the chairman and also invited the dean to visit this facility, and I invited all of you to come visit. You have an idea, you have a passion to make it happen. That's very good start. Although I just saw while passing through the Arthur Street exit, there is a engineering innovation center too. So similar kind of incubators. That's a good place to start. Okay, so that place that I showed you is called now the One Innovation Center. There are 2 or 3 different ones, and it is in the University of Louisville J.D. Nichols campus, and so that's where that is. And inflammation, being close to the university, you have interactions with a whole lot of faculty in the medical school, and hopefully we can do the entire drug development in the University of Louisville campus, and bioengineering hopefully will play a big role in moving this forward, and the goal would be to manufacture VCP ultimately within this campus and the university system. Okay, so the Inflamet team has, of course, we talked about ownership, and the University of Louisville Research Foundation is a part owner, but they have said we are not going to do much more than we have done. They have a nice office looking up to the baseball field there, but that's all. So there's a vice president who is also in charge of the business development part, and is already thinking big about what could happen and how he could get rich. So different people have different purposes in life, and mine is not the same as his, but at least we are committed to making this work. And then we also, besides Alzheimer's, what I'll be talking about, we have a couple of my former students. One was an Army major who was already had a degree in veterinary medicine, and he came and the Army gave him 3 years, told him, we'll pay your fees, but you've got to finish in 3 years. So find a mentor who would have you finished in 3 years. And so he came looking around and came to me, and I was very happy to. And he worked very hard, and he did learn microsurgical skill, and then he did xenotransplantation across species using VCP to facilitate that. I'm not going to talk about that work, but I'll just keep mentioning what else we did. And then there was one from the Indian Institute of Technology, Dr. Amod Kulkarni. He was not happy with what he was doing at the IIT, which is a very prestigious place. When he told me he would like to work with me after listening to my seminar, I said, wait a minute, I mean, you are at one of the best places for technology in India. Why would you want to come to South Africa? At that time I was in South Africa to work on this. He said, I just love your project. I was always thinking of getting molecules to the brain, because the brain has the blood-brain barrier, and you cannot do IV injection or put something in your mouth and expect it to reach the brain. Although these companies are trying to do that, and they're having a lot of side effects from trying to bombard the delicate blood-brain barrier. So I said, okay. So he told his advisor there at IIT, he's leaving, and so he started the work, and he started working on directing proteins which cannot go past the blood-brain barrier to the brain. So how did I start? As I told you, I started with not gain of function, but loss of function. So basically, yanking out genes from the virulence genes and checking whether the virus, the recombinant virus, is now less virulent than the wild-type virus. We call the original virus the wild-type virus. So that's how we started. And at that time, gene sequencing was not to the extent that it is today. And so I had to sequence large chunks of DNA. That was my first project at the National Institute of Allergy and Infectious Diseases. I don't know, some of you may have heard of National Institute of Allergy and infectious diseases. If you have not heard about that, you might have heard about the director of that, Dr. Anthony Fauci. I didn't work with him, but he was the director at that time, and he just retired in December 2022. But I worked with a member of the National Academy, Bernard Moss, who was a great scientist with a passion for science. And he was one of the very few people you could find all the time in the campus. And you could ask him questions anytime, day or night. So that's where I started. And so that whole loss of virulence brought about this whole, was really the nucleus for where we are today. And one of the genes that we discovered was VCP, or the vaccinia virus complement control protein. And the talk will be a lot about VCP, which will be the therapeutic that we'll be talking about. So this one, we sequenced it and we found that it has structural similarity, and then later we also found that it has functional similarity to the human complement proteins. So, big deal. What's the big deal? The big deal is that the virus engineered over its evolutionary period and took this large molecule and condensed it into a small, compact molecule that is VCP. And that's why it is the ones we are using. So the complement system is a powerful system in the body. It is useful to as the first line of defense against microorganisms. So if a bacteria comes in, if the body has not seen this bacteria or virus before, the complement system gets into action and it neutralizes the virus, or it ends with the bacteria gets lysed with the complement system. So that is, and amyloid plaques and tau fibrils are the hallmarks of AD pathogenesis. What does that mean? That when there is amyloid plaque in the brain or the tau fibrils in the brain, it causes Alzheimer's disease and the loss of brain function and memory and so on. So that was that. And so what's the link between the amyloid plaques and complement? The amyloid plaques are formed by a protein called A-beta peptide, or the A-beta fibrils. Now that activates the complement system. So that's the bad effect of the complement, is it damages the brain cells, the neurons, and so on. So the amyloid protein fibrils activate the complement system. And that's where we come into the picture. And that was first shown by Andrea Tenner from UC Irvine, and which then causes inflammation. So she showed that it directly causes the complement to be potentially activated. What we showed is that interaction causes inflammation in the brain. And that was a key finding. When I had a student who came from Bellarmine who started me basically on this whole project. He came in, he had just lost his grandfather to Alzheimer's disease, and he wanted a project for his dissertation. And he said, I'd like to work on Alzheimer's disease. I said, wait a minute, we've never worked on Alzheimer's disease before. And we had at that time not shown any connection between Alzheimer's disease and inflammation and Alzheimer's disease. But we were working on a complement at that time. So then anyway, he got started, and it was a lucky break that he could show for the first time in the entire world, right here in Louisville, at the University of Louisville, that the A-beta peptide causes inflammation. We had a system to check that, and it caused inflammation. And so That is part of the pathogenesis, which means causation of disease is pathogenesis. So that's how that was caused. So then we showed that the complement activation occurs by A beta fibril, and VCP, our complement inhibitor protein, abolishes that. And that was published in the Neurobiology of Aging. And we have never received such great reviews for our manuscript than from the reviewers of this. They basically said, where have you been? Why did it take so long for you to come up with this kind of a discovery? And we were just happy to address the other questions and get it published and established ourselves as the pioneers in the brain inflammation, which causes the, which is a key part of the Alzheimer's disease pathogenesis. And that's what has impressed the scientific review committee about that. So let's talk a little bit about Alzheimer's disease. You have generally heard a lot about it. And so basically, Alzheimer's disease is the most common form of dementia. So dementia can be caused by multiple different causative agents, but one of the major causes of dementia is Alzheimer's disease. And there are 6.2 million people in the US with Alzheimer's disease, and it has become global because it's not just in the United States people live longer. 20% of the world's over 100 population lives in the United States, but there are 80% living in the rest of the world. And so that it's not just a problem of the US, it's sort of all over. And that's because people are living longer than they used to. In the last century, they're living longer. Of course, some cut their life short with drug abuse or whatever, but those who do everything it takes to live long will live long. Everybody here has potential to live to 100. But is everybody going to live that much? Sure you can, but you have to do the right things to get there. And I had published an article in the local newspaper when my father turned 90. And I thought, oh, that's longevity. He has some secrets that he can share with the rest of the world. It's not just lifestyle and the diet and things like that. It's a lot. Because you got to keep your fitness here, and he had a very good memory. And of course, he then, I sort of applied the knowledge of his memory to try to understand why he has a good memory. Remember I told you about that APO? I wanted to know what his APO is. If he has good memory, is it because of that not having that APOE4? And that's what I studied after that. And so 13% of people over 65 have Alzheimer's disease or dementia. And everyone wants to live longer, but not too many want to do what it takes to live longer. It takes a lot of different things, and it's not a talk about longevity, otherwise I would have given you all the secrets that he had of living longer. So causes of dementia are many. I just talked about today about Alzheimer's disease, but be aware that that's not the only cause of dementia or cognitive decline that's talked about quite a bit. And then there is vascular dementia, where the blood circulation to the brain is not optimal. So again, my father, who lived past 100 years, used to do one thing every day. Guess what it was? To get circulation to the brain. What do you do to get circulation to the brain? He would, if that's the head, that's the feet, he would do this every day. And he didn't go to, he was a commerce graduate. He didn't go to any scientific thing. Of course, he had heard yoga people tell him about that. So he could do that, and I tried to do that by doing it against the wall so that I could keep my head down and then have the legs. And that he would do to get the circulation going. And he said that. Why did you, I would look at him down and say, why are you doing this? He said, to get my circulation to the brain. And that's very important because the carotid artery sends the circulation to the brain. And that way one can keep the brain well nourished. Although the blood does not go into the brain. Remember I told you there's a blood-brain barrier. So the blood only goes up to the blood-brain barrier but not inside the brain. If it's going, blood is going in the brain, that's not a good thing. So when the blood goes around the brain, where the blood-brain barrier is, it provides nutrition that goes inside the brain, but without, and therefore only small molecules, small amino acids and glucose and things can go into the brain itself. But that's a good way, and of course exercise is, aerobic exercise as well as all of that is very important because the brain still needs oxygen, and that's the path by which it gets there, and so that's an important thing. So just the statistics: 1 in 3 seniors dies with Alzheimer's disease or another dementia. So a decline in memory and is fairly common, and not knowing what's happening and things like that, not recognizing people can become a problem as a person. But as I said, it doesn't have to be that way. Like my father, he had sharp memory right up to his age of 98. And then he fell a couple of times, and then he was bedridden, but then still had good memory. When in his 100th year, his granddaughter wanted to check his memory. So, and there was Putin visiting India at that time, and my father was in this thing. So she said, "Grandpa, do you remember the name, the first name of Putin? He's visiting India." Without a delay, my father said, "Vladimir." and she was totally, because he was in his bed since he fell, but his memory was still pretty sound. And so but that memory is partly due to what he did in his lifetime, as well as what he did along the time. Okay, more than 6 million, we already went over that in America, and then there are probably another 5 to 6 times. He had excused himself, he was going to go to a meeting at 1 o'clock, but well, what's the time, by the way?
It is quarter till 1.
Quarter till 1, okay.
Yeah.
Okay, so the hippocampus and entorhinal cortex is where the memory is, and so that's where we target our protein. So how do you know that there is Alzheimer's? 100 years ago, when Alzheimer's discovered the disease, they didn't have a PET scan. There were not enough good engineers then. So the only way he would know it by talking to the person, and then after the person is dead, he would get the brain, and then he would come up with the plaques. So what you see here in an amyloid brain are those plaques. So the brain, the normal brain, it does not have has those plaques, whereas an amyloid brain has the plaques. So PET scanning is very good for monitoring how the treatment is working. Because if you are targeting amyloid, which is a good target, although I have problems with that, but I won't try to discuss those. But basically, as you can see on the right side, after 1 year, it's dose-dependent reduction in the amyloid plaques, which is a good thing, and that is from a drug developed by Biogen. So PET scanners, again, a remarkable engineering feat of doing it, the diagnosis and so on, and that's the kind of scan you get. Risk factor, as I said, age is the biggest risk factor, and we have already gone over the statistics. Gender, unfortunately, females have a greater possibility of Alzheimer's, and one can look into the family history. So what predisposes? What is the genetic predisposition to Alzheimer's disease? As I said, the ApoE gene is, and the ApoE4 allele is the biggest one. Going back to again, this was a classic paper, because here we have given the entire technology in this paper on how to sequence the gene, the ApoE gene, and to find out whether a person has ApoE4 or ApoE3 allele. And the conclusion from that was, if you have the ApoE3 or ApoE2, then you have a diminished incidence of Alzheimer's disease or dementia, and that can contribute to longevity, because very few people who have lost memory are able to take care of themselves and live long. So the two major factors that contribute to the pathogenesis are the neurofibrillary tangles, NFT, and the beta amyloid protein that brings about the amyloid accumulation in the brain. So Biogen targeted the amyloid protein. That's a good target, because as you can see from their PET scans, they were reducing that. But the way they deliver it is by IV injection. When you inject by IV, you have to go past the blood-brain barrier. To go past the blood-brain barrier, you got to put a whole lot of the drug infusion. So you have to do it every 3 weeks to do that. And then it's only good for mild cognitive impairment. And that's why the insurance companies would not pay for this treatment, because it had the side effects. So these are some of the problems with the ADUHELM, as I mentioned. The blood-brain barrier, although it decides what molecules go in, larger molecules are kept out, it's still delicate. So our approach was targeting the complement system in the brain to prevent the inflammation. So as we talked earlier, the amyloid plaque activates the complement system. The complement system then releases chemotactic factors that attract an inflammatory response of the microglia, the macrophages in the brain, and the astrocytes, which then cause the neurodegeneration. And so blocking the complement system in the brain is our target, which is downstream of the other targets, but it's still worthwhile. So this is the complement system. I won't go over it, but basically it is activated by the presence of antibody binding to the antigen and or certain surface molecules, and then that activates the complement system. The purpose of that primarily is to protect the body against foreign bacteria and infectious agents and so on, and by inducing phagocytosis and lysis of the bacterial cells. The complement system is a double-edged sword. What I told you about in the previous slide were the good effects of complement. But then it's a powerful system. If it is not controlled with regulatory molecules, what's going to happen? It's going to damage the tissue. So there'll be a self-destructive effect So on one side, it will attack the infection, on the other side, it will cause in the inflammation. So it has those two sides. So, and to control the inflammation, and there are several inflammatory diseases that you may have heard of, arthritis and other inflammatory diseases in the body, and so it's important to control inflammation. And what is the relation between Alzheimer's disease and complement? As we were saying, the complement molecules bind to the amyloid plaque, and that then activates the complement system, that then activates the inflammatory response, that then causes the neurodegeneration. So best thing is to try to down-modulate that. So VCP is a candidate for vaccinia virus complement control protein that I discovered back in NIH, which we published in Nature in 1988. It is made up of 4 repeat structures, repeat molecules. And those repeat molecules gives rise to this compact structure on the left side. You have those 4 boxes there. That's the VCP. And it also has a heparin binding site. And each is made up of 60 amino acids. Compare that to what is the human counterpart to which it is similar. It's a large molecule, it's 450. Difficult to produce, difficult to fold, difficult to administer, and so on. So the advantage of a small molecule that like VCP to control complement is quite a lot. So we have done a whole lot of basic research on the structure, on the stability, freeze thawing. It's temperature resistant. You can take the protein right up to 95 degrees, and nature has constructed a really robust molecule that still does not lose its functional activity. And then we did a whole lot of preclinical in different disease models and showed that it does the job of taking care of the immunity, of the inflammation. And we developed a model, a rodent model, and we were able to show that the actual functionality, memory, improves in Alzheimer's transgenic mice. And so it almost comes to the level of the non-transgenic mice which don't have Alzheimer's. So that is the advantage of this system. We then expressed it in the Pichia pastoris system, another genetic engineering, took that gene and put it in an expression vector which has its own promoter and so on, and then grow the yeast in large quantities, and it goes out into the medium, and then you harvest it from the medium and then purify it because of its heparin binding activity, and then it's ready to be administered and checked out. Of course, you need large quantities of that. We tried that in a baboon and showed that it can knock out the complement system, but then we needed to go to humans and we needed a large quantity. So we started scaling up. The first step for scaling up was a 14-liter biofermenter that we used, and where we got milligram quantities, but that's still not enough. We need gram quantities. And so we approached White Oak engineers, the local engineers in the States. White Oak stands for Virginia, Indiana, Tennessee, Ohio, and Kentucky. And they have designed this system to produce it, gram quantities. And we were waiting for more funding to fund this particular thing so that we can get large quantities of that. So that's their organization chart. There's a lot of engineers involved there. So there's a whole lot of promise from VCP. These were some of the students who were involved in this project. As I mentioned, James Daly was really the first one. He was not happy with just getting a PhD. He went to the Brandeis Law School and got a JD so that he could become a patent attorney, and now he's the senior counsel at Dow Chemicals in Indianapolis in their patent department. Nirvana Pillai did some of the transgenic work showing that VCP actually has behavior things. Then Johannes Nirvana was from South Africa. Gabriel Mariam was from originally from Eritrea. Now he was involved in mutating the gene so that it is more active than the original natural VCP. And then Scott Smith did a whole lot of stability work on the VCP, and now he's a faculty at Vanderbilt with several grants and very successful, and came from Pennsylvania, farm country where nobody went to school, and he came here, got his PhD, then got an MD, then did a fellowship in this thing, and now and he has discovered the dengue vaccine. So, as they say in Canada, there's a bank that says, when you succeed, we succeed, which means when the students succeed, we succeed. And so that was, when I hear these stories of their success, I feel good that the education they got was benefited them in their career, and they made good use out of it, most importantly. There are students who came and went to music and things like that. That's fine, music is great, but that's not what I thought they would go into. And then there was a postdoc from Nigeria, Odutela Odunura, and he was a postdoc, and he did the site-specific mutations to engineer the gene into that. So none of this comes without adequate funding, and it never comes in time. My postdoctoral work was funded by at the NIID, NIH, plenty of funding. There the problem is not how to find money, the problem is how to spend the money. And so end of the year they would come and say, hey, anybody wants any instruments? So we have a couple million lying around. Okay, that doesn't happen in real life outside of the NIH system. And then the Kentucky Spinal Cord Head Injury Trust in Kentucky helped with the head injury and the spinal cord injury research that we did. And of course, we could do some research on Alzheimer's due to that. Because another thing is, another risk factor is head injury. If one has had a head injury in the childhood or something, that can progress to Alzheimer's disease. So that is another way one could get Alzheimer's disease. And then we are now pretty much about to hear about the actual money coming in from the National Institute of Aging, which has which is involved in research related to aging population, which is very big and very powerful in terms of their votes and so on. And so the National Institute of Aging works on that, and so we are about to get a funding from that. So thank you very much, and I'd be happy to take any questions. I'm sorry I went beyond, I didn't expect to. That's why I was trying to come early to get this going. There's always a problem with putting in the things no matter where, it's universal. So that I tried to get over earlier. But yeah, feel free to fire any questions that you may have.
So can I go first, and then unfortunately I have to be the student. Your company Implement, how is that funded? Is it from, is it commercial? Are you commercializing a product and funds come in for R&D? Do you have current SBIR funding to support that?
Very good question. So far it has been funded by myself and by the Vice President. So this is our first SBIR that will fund the future. In fact, we are going to subcontract some of the animal work to a company called Psychogenetic, because we did our studies and we are pretty happy and convinced that what we have will be a good treatment for Alzheimer's. But not cure, but treat Alzheimer's.
And your SBR is a phase 1 that's awarded or about to be awarded?
It is. It has been funded. It was waiting for the omnibus to be passed. So that has happened. And now we should get, and we are doing now the just-in-time documents and all kinds of paperwork, bureaucratic things. And today, in fact, in the morning, the first one thing I did was that. And so after that, the money will come 1st of February.
It's a phase 1, you said?
Phase 1, yes. But those the local banking bankers, investment bankers who don't know about this story, they said, hey, can we pay the money? I said, wait a minute, we need to do some milestones first. And that's what this phase 1 will allow us to do. Milestones in the sense to look at the feasibility of doing a long-term this thing. So we are trying to take it one thing at a time. Once this comes, so that lab that we are getting, phycogenics, to do the behavior studies, will do it independently of what we have. So we have plenty of data. We have convincing data, but it's better that there is some overlapping data from an independent lab, and that's where we are.
Thank you.
Anybody else?
Yeah, feel free. The only silly question is one that is not asked.
I guess I have a question.
Yes.
About delivery of these treatments,
Yes.
these drugs. So
Yes.
I'm trying to think back to my physiology on the blood-brain barrier. Is it a matter of attaching them to something that already goes in, or making them small enough to where they can pass through, or?
No, that's a very good question, and that is the question on which we spend a whole lot of time in South Africa. Because the way it is delivered by the FDA approved is by IV injection. We don't know what proprietary thing, what makes it go through that. But they are basically causing side effects from bombarding the blood-brain barrier to get their product in the brain. So what we are doing is intranasal delivery. I showed the brain map there, and next to the region that protects the memory is the olfactory lobe. So when you deliver something through the intranasal route, it diffuses into that area, and that's how we have already tested that. But we want, again, independent testing of that. So, but you're right, that has been the major questions, because you can send small molecules, and they have been tried and failed. They didn't work. Lilly and big pharma, we're just little guys in Louisville, but the big pharma had already done this. But they didn't have the desired result of protecting memory. Whereas we have tried and we have protected memory. Now we just need to actually produce it on mass scale. That's where I want closer interaction with the future bioengineers that I see in front of me, and work with them to try to produce this. Plus, even the delivery part, just dropping something in the nasal drops, that won't go. You got to really push it hard under pressure so that it reaches the olfactory lobe, and then there is enough bioavailable protein to diffuse in the rest of the brain. At least more in the limbic region of the brain.
Oh, over.
Okay.
So in terms of manufacturing, since you already have a quality control, I'm assuming all of your manufacturing is taking place under QMS, or is that the goal?
The manufacture, so far, we have only done up to 14 liters. So we have milligram amounts. That doesn't last too long.
Yeah.
So in fact, I was very impressed when I went to Oslo. There was this big truck, and the truck stopped in front of a bar, and then he unwound the tubing, and then he took the tubing there, so that the beer from that big thing, not in bottles, went in there. So that's my dream, to have enough so that we can deliver to the hospitals large quantities of this hospitals or even homes in that amount. So that White engineers were involved in that, but that was like 3-4 years ago, and we were supposed to find some big money for that. So our phase 2 will be to try to manufacture it, get a lot of money, and then, so ultimately we have to raise up to $100 million and produce a lot of protein. But I want that $100 million spent here in, in this place.
So it needs to come under GMP, and then it
Yes.
needs to be all under
Yes.
UMS, all of that standards.
So yes, we have done everything under GLP, but that's not, it has to be, that's not going to take us too far. So we have to go through that process of GMP certification, and we have to show that we can reproducibly produce that protein to the FDA in quantities enough to distribute it to the population. So there are a whole lot of challenges, and, and it's not up to former academicians to be trying to deal with those challenges, and that's where it's all engineering now, bioengineering that's going to collaborate with, will help us move forward.
Again, my background was in chemical engineering, and so, reactors and things like that. But on the flip side, you talked about diffusion related, and you were also talking about pressure. So when you hit any surface with pressure, it really doesn't matter, because diffusion is going to be the limiting step. Are you encapsulating something? I know you collaborate with Dr. Sufan Chen, who
Yes.
encapsulates it.
Yes.
So are you looking at encapsulation so that it makes it easier to go through, hydrophilic, hydrophobic
Yes.
barriers? And
What I was working with Sufan Chin, and we published that work in Nanomedicine, was to get the protein, to get the ATP inside cells. Now this one, we are not trying to get inside cells, because the complement system is outside the cells, and that's where we want to block the complement system. So I think we may not need, but we have thought about PEG, pegylating it, and other things, and we could probably pursue that. But what we are trying 2 tracks there. One track is to make this more potent by mutations. We already made 3 mutations, and it is now 64 more potent, so that lesser quantity can achieve the same result.
So in terms of your delivery, why do you need it under pressure? So that was kind of my, it didn't make sense, because if diffusion is usually the slowest step.
Yes, but there are others who are working on that, and they are also doing it under pressure, because we are not so much worried about the blood-brain barrier, because we are not giving it IV.
Yep.
We are just trying to deliver it to the olfactory lobe, from which it diffuses. But even for the olfactory lobe to absorb enough quantity so that it diffuses enough, I mean, what we gave was few micrograms, and it worked. But that doesn't mean it might work in humans.
And also, how long did it work in terms of, it's one thing to give a dose
Oh,
once a day, so
okay.
what is the dosage that's required?
Very good question. Because what happened is, what we gave within the first 6 months prevented the Alzheimer's-like pathology for 2 years.
Okay.
So we don't know, don't ask me how, but it seems to stick around there and do the job. So, but, and based on that, we don't have to worry about the time. Although in our studies we are going to keep doing this a good number of times. And this, if we ever succeed, will be better, because then it doesn't have to be delivered in the hospital. The current Alzheimer FDA-approved treatment has to be delivered in the hospital. The person has to go and IV, sit there for 1 hour while the thing has been infused. This the person can do under pressure, and there's a company in Lexington that does intranasal things, but it preferably at a high pressure, so that it larger quantity gets in and then stays there. And so the person can do, like they give nasal sprays for asthma, allergies, or hay fever, same thing like that. So this will be a lot better. And secondly, as I said, This is not a cure, and it will be for those who are predisposed to Alzheimer's, not trying to treat people who already have Alzheimer's. It is for those who have, and why will they go to sit and get
So less
IV?
invasive option for predisposed, and then you also have to figure out how you're gonna get reimbursed by, when you become.
Very good question. That's their problem, not ours. Because their treatment costs $50,000 per year per patient. Oh, did you have a question?
Sorry.
I thought you were raising it. Okay.
No, they got classes.
Okay.
We can end the seminar, we can talk later, it's
Okay.
not an issue. Because I
Oh,
think I have classes.
Well, I had a question about the dosing too, is the schedule, so are you all thinking it's going to be done every day to maintain
Yes.
that? Okay.
Yes, because it goes back to that question, how much, it's very hard to estimate how much has gone in.
Right.
You don't want to break the skull to find out how much is there. So you just have the person put it as much, and so there'll be a trial of those who will do the squeezing, and then follow them up, and then follow those who have not done the nasal drops, and so it'll be easy to administer and get it to the brain where it's needed. And so, so dosing, those will still keep coming. We can't really dose it because we are not giving it IV, we are not giving it orally. So this is a little bit
Yeah.
quasi-science in a way. It's more an art as to how many times. And that's why the sprays have to be really very strong sprays. You know, there's some of those flimsy bottles, and you press them,
Right.
and it doesn't do much. So that will be an engineering feat to have something that will send it much more better.
You said that this molecule stays very long, and you also want to give it repeated, daily dosage. Have you looked at toxicity in terms of accumulation?
Very good question, because every time we ask is, does this elicit an immune response and get inactivated in the process? No. Why not? Because it is very similar to the human counterparts. Because the human body makes those complement control proteins too, except they are large for one thing, and the second thing is the molecules that the humans have would be much harder to produce, and we have to introduce a lot more to achieve the same result.
So there's no cytotoxicity to those cells
No.
up in the nasal cavity?
We have gone right up to baboons. We have gone through rodent models, and we had the baboon study, and we were able to knock out the complement system of the entire, that was given IV, but that was just experimental to see if we can knock out the complement system completely, and it did that, except we didn't have enough to keep keep that going for a long time. This is a little different, and so this way we should be able to do that.
Anybody else? Well, thank you very much.
you're welcome.
And I would clap, except if I let go of this iPad, it's gonna fall down.
No, that's fine. Thanks for the attention, and
Yep, now I can do it.
happy to be here. I never saw